Assalamu alaikum friends, I'm OMAMA with a new video in which I will be discussing antibodies, its types, how they are formed, and what's the mechanism by which they destroy the antigens. My channel name is OMAMA SHAH MEDICOS. If you like my videos, must subscribe to my channel so that you should get notification of my new uploaded videos. So friends, we will start from the introduction of antibodies. Here you can see the structure of antibody. This antibody is immunoglobulin, as immunoglobulin is a protein, so antibody is protein in nature. It has 4 polypeptide chain, 2 heavy and 2 light. Highlighted in green is the heavy chain, while highlighted in light blue is the light chain. Both of these chains are joined together by a disulfide bridge and form a Y-shaped structure. This Y-shaped structure is basically called as antibody. The amino sequence in the tip of this Y varies greatly in different antibodies, and this specifies the antibody for a specific antigen. Now coming towards the classification of antibodies. Antibodies are classified into IgM. Whenever an antigen enters the cell, the primary response is given by IgM antibody. IgM has 10 binding sites. It is therefore the major antibody present in acute infection. IgA, whenever there is excretions in the body, whether it may be in the form of tears or mucus excretion from the GIT tract or breast milk or saliva, there will be production of IgA antibody, immunoglobulin A. IgD. IgD matures the beta cell. IgD regulates the beta cell. When an antigen enters the cell, it is IgD that signals the beta cell to be active to produce the antibodies. IgE. IgE antibody will be present in allergic conditions. IgG. Whenever there is chronic infection, the IgG antibody will be high in concentration. IgG is bivalent such that it has 2 binding sites. Now we will be studying how antibodies are formed. So, whenever an antigen enters the body, it is first presented to B lymphocytes and T cells. The T cells activate T helper cells, while the B lymphocytes enlarge and take on the appearance of lymphoblasts. Lymphoblasts further differentiate and form plasmablasts. Plasmablast gets mature and form mature plasma cells. The mature plasma cells in turn give rise to antibodies. If I mention the sequence, first produces the B lymphocytes. It enlarge and form lymphoblast. Lymphoblast differentiate into plasmablast. Plasmablast get mature and give rise to antibodies. In conclusion, antibodies are formed from B lymphocytes, not from T cells. Antibodies are formed from B lymphocytes. So this was all about formation of antibodies, how antibodies are formed. Now coming towards the regions of antibodies. Antibodies has 2 regions. One is the variable while the other one is constant. With variable you can see V written, while with constant the C is written. As I discussed you earlier, that the antibody has 4 polypeptide chains, 2 heavy and 2 light. The heavy is highlighted in green while the light is highlighted in blue. This region is called as Fab region. Fab stands for the antigen binding fragment, such that this region will recognize the antigen. While FC region stands for crystallization fragment, and this region will promote removal of antigen. By which mechanism the antibody will remove the antigen, it will be determined by FC region, while the FAB region will bind to a specific antigen. The variable region gives the antibody its specificity for binding antigen, while the constant region determines the mechanism used to destroy the antigen, as I discussed a few minutes earlier. So this was all about the regions. So friends, now we will be studying the mechanism by which the antibodies will basically kill the antigens. It will destroy the antigens. So there are 2 mechanisms by which antibodies will either kill or destroy the foreign particle, which are basically called as the antigen. One is the direct mechanism, while the other is the complement system. We are going to discuss each of them one by one in detail. So let's start from the direct mechanism, by which the antibody is strong enough and it will directly kill the foreign particle or the invader, you may say. So let's start without delay. Direct action of antibodies. In direct mechanism, the antibodies are potent. They are strong enough to attack the antigens directly, right? And they do it by some processes that are, it may be through agglutination. In agglutination, the antigens are made into clumps and then removed from the body. By doing this, the antibodies basically save time, and it is, in other words, very much difficult for antibody to first kill one antigen, then other, and then the next one. So what it do, it put all the antigens together, made it into clump, and then together it destroy, it kill all the antigens by phagocytic process, right? And in this way the antigens are removed. So first is this step, agglutination. The second is precipitation. The precipitation process is basically used for diagnosis. For example, if we have a blood sample and we want to diagnose if there are antibodies present or not, for example of tuberculosis. So what we will do, we will take a sample of tuberculosis and then we will put that antigen that causes tuberculosis, the bacteria that causes tuberculosis, we will put that in that sample, blood sample. If they form a complex, the antibody and the antigen, and precipitate, it will indicate that the blood sample has antibodies against tuberculosis. So basically this precipitation process is used for the diagnosis purposes, right? So if antigen antibody complex will form, it will precipitate and it will show us that the specific antibody for specific antigen is present in the specific blood sample, right? The next one is the lysis. what do the antibodies do in lysis? The antibodies attack the cell membrane and it causes a rupture of the cell membrane, right? By doing this, all the solute enters the cell and osmotic lysis takes place. So the antigen, it may be killed through the lysis, means the antibody and the lysis will attack the cell membrane of the antigen. The last one is the neutralization. Basically, not all the surfaces or not all the antigen basically infect a cell. There is some portion which causes infection to the cell and which is harmful for the cell. So the antibody in neutralization cover that site, right? So if it cover that site, so that antigen will then not affect the cell. So in neutralization, this technique is used by the antibody, and by these four processes, agglutination, precipitation, lysis and neutralization, the body is remains protected by the antibodies from the attack of the antigens. Now we will be studying the complement system for antibody action. So complement is a collective term that describes a system of about 20 proteins. But here the principal actors in this system are 11 proteins, C1 to C9, B and D. This complement system initiates whenever the cell is attacked by an antigen and it binds with the antibody, forming antigen-antibody complex. Whenever this complex is formed, what happens, there is a specific site on the constant portion of the antibody. That portion becomes uncovered and it in turn binds to the C1 molecule of this complement system. This in turn stimulates the sequential reactions, and at the end we get products. That products are responsible for different functions. Here you can see the C3b, it is responsible for the opsonization of bacteria. Opsonization is basically the tagging of bacteria. This C3b identifies that some foreign particle has come into the cell. It will stimulate or attract the phagocytic cell to come and kill this bacteria, phagocytize this bacteria. The other product you can see is the C5, B6, 7, 8, 9. This product lyses the cell, means it ruptures the cell membrane as I discussed you previously. The product C5a, it do the chemotaxis, means it will secrete some chemicals that will attract the phagocytic cells such as mast cell or the other phagocytic cell eosinophils, and it will then phagocytize that particular antigen. The C5a, C3a and C4a, as you can see over here, is also responsible for the activation of the mast and basophil cells. When the mast and basophil cell activates, it releases histamine, heparin, and several other substances into the local fluids. These substances in turn cause increased local blood flow, increased leakage of fluid and plasma protein into the tissue, and other local reactions that help inactivate or immobilize the antigenic agents. These same factors play a major role in inflammation. So this was all about the complement system for antibody action. Now we will have some major questions regarding this. Answer that question in the comment box. Coming towards the question of the topic. Number one question is, Which protein is responsible for opsonization? Number 2, which protein is responsible for lysis of antigen? Number 3, C5a initiates which action? Which antibody gives primary response to an antigen? And the last one, which portion of antibody initiates the complement system? If you know any one of the question answers, write it in the comment box. At the end, I would like to say that subscribe must to my channel for new uploaded videos. Thanks for watching. Allah Hafiz.